Welcome to the 16th installment of the PhenoTips Speaker Series. If you're new to the webinar series, then we're happy that you've joined us the first time, and a warm welcome to those who are returning and tune in regularly. I'm your host, Dr. Pavel Buchkovic. I'm the Chief Operating Officer and VP of Scientific and Medical Affairs at PhenoTips, a software and service that makes genetic professionals' workflows easy and intuitive, and incorporates features such as pedigree drawing, standardized symptom capture using the human phenotype ontology, and insights such as differential diagnosis and gene suggestions. Since electronic health records aren't built for genetics, PhenoTips fills the gaps by providing a complete suite for genetic medicine. In light of the pandemic, PhenoTips is sponsoring this speaker series. The format of the webinar today will be a 45-minute presentation, followed by questions from the audience until the top of the hour. You'll be able to submit your questions throughout the presentation in Zoom's Q&A box. Our speaker today is Dr. Anna Cohen. She is an ABMGG certified laboratory geneticist, focusing on the analysis and interpretation of genomic variants detection among pediatric rare disease patients. Dr. Cohen received her PhD from the University of British Columbia, focused on the power of deep phenotyping, and described the first two patients with embryonic ectoderm development related overgrowth, also known as Cohen-Gibson syndrome. Dr. Cohen is an assistant professor of pathology at the University of Missouri, Kansas City School of Medicine, and an associate director of molecular genetics at Children's Mercy in Kansas City. As part of the Genomic Answers for Kids research program at Children's Mercy Research Institute in Kansas City, Dr. Cohen is currently investigating additional strategies for diagnostic success in rare diseases. The title of today's presentation is Children's Mercy on Ending Diagnostic Odysseys. The stage is yours. Please take it away.
Thank you so much for that kind introduction, and to everyone listening, wherever you might be, good morning, good afternoon, good evening. Thank you for joining us. So I'm very excited today to share with you the work that we're doing here at the Children's Mercy Research Institute, and particularly within our program, Genomic Answers for Kids. And of course, we will be highlighting how PhenoTips has been helping do this work. And I will start, given that a lot of you might not be familiar with our center, giving you a little bit of an overview of where we are. And so the Children's Mercy Hospital is an expert in pediatric care for the wide region that covers Kansas, Missouri, and also neighboring states. It is a nonprofit independent children's hospital that has been around for a while, since 1897. It has almost 8,000 employees, and it has over 40 pediatric subspecialty areas with almost 800 specialists, of course, many nurses, allied health professionals, and a very large number of volunteers as well. They are at the highest level for pediatric trauma care, and of course, also for neonatal intensive care. And they are repeatedly ranked amongst the best children's hospitals in the country. These are just sort of an overview of what the health system was seeing, and bear in mind, of course, these are pre-COVID numbers. So if we didn't have a pandemic, this is sort of the volume that the hospital was seeing, with over 400,000 outpatient visits a year, over 200,000 ER or inpatient visits. Really, as I mentioned, it's a big hub for all the pediatric care in the region. And if you don't know where Kansas City is, I will show you here, because I myself didn't know before I came here. And it's smack right in the middle of the United States, as you can see. And also, to add some little peculiarity, Kansas City, the city, sits right at the border between Kansas state and Missouri state, and actually part of the city is on one state and the other is on another part. And the hospital system is actually divided in multiple regional centers, and with the star here, you can see Adele Hall, which is the main campus, which is where we are located, but there are centers in other regions that are trying to serve the pediatric populations that are not close to the main hospital. And we are very lucky because just in late 2020, the hospital opened their brand new research tower, which is where we are now currently doing our awesome work. And I just wanted to share that if you look at the research tower at night, you can actually see these different patterns of lights, which are purposefully constructed to symbolize what used to be sequencing gels back in the day. So when they used to do Sanger sequencing manually and then run that on gels to find out the sequence. And these actually represent 4 patients who had who got answers for their medical care through sequencing at Children's Mercy. So now a little bit about what our program is doing. So what is Genomic Answers for Kids? So this is really a massive undertaking led by Dr. Tomi Pastinen here at Children's Mercy. And it is meant to be the first of its kind pediatric data repository that really wants to facilitate the search for answers and novel treatments for all pediatric disorders. And our goal is to collect data and health information for 30,000 children and their families over 7 years. So eventually having a database of nearly 100,000 genomes. Why are we working on rare diseases? So there are, at this time, over 6,700 genetic inherited diseases that have been characterized. They affect 1 in 30 children in the United States alone, which causes 1 in 6 children's hospital admissions and 1 in 5 infant deaths. And the diagnostic testing that is currently offered and has sort of become the standard of care is symptom-driven clinical exome sequencing, but it only resolves about 25 to 35% of cases. And so a lot of these children remain without an answer to what is causing them sick, which is often really important to guide their care. As I mentioned, the goal is to eventually have 30,000 patients recruited and 100,000 genomes, and this is where we are to date. So we're over 3,000 patients, which together with their family members that have joined the study, we're at over 8,000. That's over 5,700 exomes, almost 3,000 short read genomes. And another thing that we are doing and innovating in this way is we're at over 750 long read genomes, high fidelity, which we have established a partnership with PacBio to do this. And it's really showing really promising results, as I will mention later in the talk. So this might look like somewhat of an overwhelming figure. Don't worry, you don't need to fully understand every part of it, but really what I'm sharing this with you for is to show that we are sort of attacking this quest to diagnose our patients from multiple different directions. And the first column on the left here represents that we're actually using different sequencing strategies. So as I mentioned in the previous slide, we're still doing exome sequencing, which is considered the standard sequencing method for these patients, but we have explored multiple methods of genome sequencing as well. And of course, a lot of our efforts go into making sure that we are looking at the correct variation in this sequencing. So controlling for technical noise and artifacts, and making sure we're filtering against anything that would be highly common in the wider population, where we make use, of course, of the huge efforts in public databases that are out there. And then we need to filter further with clinical information to figure out which rare variation detected in these patients might actually be giving us a clue to what is making that specific patient sick. And for that, we incorporate a lot of information, including, of course, inheritance pattern. So meaning, is there a family history? What do we expect this disorder to be? Is it something completely new? Was it inherited from a family member? And that would point us towards a mechanism of disease that could be autosomal dominant, autosomal recessive, or X-linked, for example. We have also explored some computational tools that help prioritize amongst the rare variants for each patient. And specifically, we've used two tools that are publicly available, and they're called Exomizer and Emily. And these tools, what they do is they take the basic characteristics of the variant, as well as explore a lot of the phenotypes that are annotated for the patients. And I will explore more of what that means. And combining all this information, it helps sort of give a score to what the most likely rare variant might be for the patients. And all of this is, of course, a very large undertaking, and it involves a lot of people. And we have assembled data on the first 1,083 patients, and we describe the strategy in detail in this paper. The preprint is available now at medRxiv, as you see here, and you can see the large author list represents just how large of an undertaking this is and the teamwork involved. And it should be available online within about a month, as it has been accepted by Genetics and Medicine as well. So if you are wanting to know more about our strategy and exactly how we've explored the different sequencing methods, as well as these computational tools to help us prioritize variants, I highly recommend you have a look at this. Now, one very important thing that I wanted to mention is part of our program is that we have a commitment to data sharing. So, of course, all information is de-identified and you cannot link it back to the patient. However, we find that it's really important to share this with the wider community, because this is really how we believe we can get to faster diagnosis by joining forces with other groups around the world. And as I mentioned, we have explored a lot of sequencing through PacBio with high fidelity genome sequencing. And we have used our study to understand what variation coming out of this sequencing might be noise versus real variation. And after all that filtering, we have curated a list of recurrent variants and their frequencies from over 1,000 alleles, and this is publicly available so that other groups that are doing the same sequencing method may check their results against ours. Now, as I mentioned earlier, and this is exactly where PhenoTips comes into play, when we are trying to solve these diagnostic cases, it is really important to collect additional data. So it is great to have multiple sequencing strategies, but of course we can't really do anything without sequencing without having the additional data. So the two main things to collect are personal and family history. And these are best captured in pedigrees. So really it's a family tree that is annotated as to who is affected, when, what features they have. And then to annotate those features, we collect detailed phenotypic information. We have a very clinically diverse cohort, so we really have a very broad recruitment of accepting any child that has any disorder that affects any part of their body that is suspected to be of genetic origin. And because of this diversity and because of how broad the recruitment is, it is really imperative that we have some sort of universal coding for these phenotypic features, and that's where HPO terms come into play. And I expect most of you to be familiar, but just in case you aren't, HPO terms stand for Human Phenotype Ontology. These were developed to have a standard vocabulary of these phenotypic abnormalities seen in human disease. They currently have over 13,000 terms that have been annotated and over 156,000 annotations to inherited human diseases. Each term independently describes a clinical abnormality, and the term can be really general, sort of just saying there's an abnormality of the head, or it can be really specific, whereas that abnormality is actually macrocephaly due to hydrocephalus, for example. And what makes this really special is that it's not a simple system where you would say, for example, abnormality of the head, and then within it, there's the option of macrocephaly, which is large head. Actually, macrocephaly can link back to abnormality of the head, or it could link back to overgrowth. So each term can actually be related to multiple parent terms. So it's really a wide net that connects all this vocabulary. So as I was saying, for this specific example, we have a phenotypic abnormality that happens to affect head and neck. And then within that, it's affecting specifically the head. And then it's affecting specifically the skull size. And then within the skull size, you can think undergrowth or overgrowth. And then for overgrowth, which is macrocephaly, there's multiple types. And why is this so important? So even if you're thinking macrocephaly seems very straightforward, but really when you look at the synonyms here, you can see there's multiple ways of saying that someone has a large head. And so this was really what led this group to recognize that there needs to be a standardized way to make sure everyone is saying the same thing in the same manner. And as I mentioned, it's not only the actual phenotypes that are annotated. Each of these phenotypes is then associated with diseases and genes. And if you see here, the term macrocephaly is linked to over 500 disease associations and over 400 gene associations. So again, thinking of that very wide network that keeps getting wider, this is really a very powerful way to annotate the features in these patients and to also pull information that will link to the genetic data. So what does this look like and how does PhenoTips help us? Well, PhenoTips, which I just took a screenshot for one of our patients, is helping us pull all of this together in a way that is integrated and easy to use for everyone. And not just that, it can actually be, it supports the idea that this is done by a team. So for example, at the top, you can have any of our recruitment team fill out the patient identifiers and basic information on the patient. Then we can have a clinical expert fill out the indication for referral and phenotypic features for different individuals in the family, as well as, of course, our a very powerful tool to draw the pedigree for the family, which is extremely helpful. And you can annotate each of the patients individually for what features they have. And all of this, of course, is connected with HPO terms that are coding those features, as well as it can all be connected with genotype information. And for the genotype information, one step further is that you can annotate whether this is truly like a causative variant that you have found, or if it's just a candidate variant, and what is the suspicion. And this can be annotated in multiple ways. And again, this part is typically populated by the expert analysts, but it's really a teamwork. And another aspect that we really like that just shows how easy PhenoTips is to use is a lot of our patients, of course, are seen in the clinic and might undergo clinical testing first before deciding whether to enroll in our research program. And a large part of the clinical team also uses PhenoTips to capture the clinical features and the family history, and then that can easily be transferred over to our research repository, which is really smooth and makes everyone's lives easier to capture all this information. So if I were to focus in here just to really show how much detail you can get, you can really annotate all the different family links, and you can annotate whether they're affected or unaffected. When you click on each of these, you would see the specific features, and you can add notes manually as well for each individual. And one additional layer that is great is that all of this that I've just shown you are screenshots for what we are using internally. But again, going back to our commitment to data sharing, we have managed to create a web cloud-hosted version of our studies PhenoTips that is completely de-identified, but still captures all the phenotypic information and the pedigrees and the rare variants that are associated with those, so that we can share with the wider research community. And we are really excited about this because we feel that, as I mentioned before, sharing genetic variation is great, but without the context of where that variation was discovered, it is not as applicable to others. And we really hope that this sharing will help accelerate more diagnosis for our patients by having additional researchers recognize similar combinations of genes and features in their own cohorts. So I hope I was able to give you sort of an overview of what we're doing and how PhenoTips is helpful. And now I'm actually going to switch to give you some case presentations for some of the diagnostic odysseys that we were able to solve. So I'll start with patient one. This was a patient that had an uncomplicated pregnancy and was delivered at 39 weeks gestation by C-section due to macrosomia, which is a large head as well. Evaluation at 7 months showed macrosomia, macrocephaly, difficulty feeding, ventricular septal defect, developmental delay, dysmorphic features, and global hypotonia. Family history in this case was remarkable for a sister that had a similar history. Interestingly, she was older and she was doing pretty well at 6 years old. The original testing for this family had been undertaken in 2018 and was negative with no compelling variant, not even variant of uncertain significance. So here we have a pedigree of the family, and as I mentioned, when you click on each node, you can see exactly which features are assigned to each individual, and you will see On the left here is the patient that I was introducing, annotated with the different features. And you can see on the right here at the bottom is his affected sister, which actually had a few more features, but the primary features are matching to her brother. And if you notice as well, if you look up, dad's is also noted to have macrocephaly and pectus abnormality. So as I mentioned, they had received clinical testing in 2018 and it had been negative. When redoing analysis within the context of the Genomic for Kids program, we actually came across this interesting variant. It is highlighted here in the center. It is a frame shift variant, meaning that it introduces a nucleotide and causes a shift in the reading frame and changes what the protein is from that point onwards. And this frame shift occurs in this gene ZBTB7A. And you can see as well that it is present in the proband. It is present in dad and the affected sibling, but is absent in mom. So it is tracking with family history. You can see that this change is happening in exon 2, but exon 1 was non-coding. So it's really the first coding exon, and it's actually a relatively small gene. This gene was not described in OMIM as a known disease gene. So OMIM is a great repository of information for that. The variants, this variant specifically was completely absent from population databases. And this gene is constrained against variation. And for us to say that, we use the NOMAD database constraint scores, where they look at the gene and they calculate based on the size alone, how many of these different types of variants would you be expected to see based on just random mutation rate, and then whether you're seeing more or less variation than expected. And here you can see the scores. The PLI at the bottom typically associates with loss of function variance, which is what we have here with a frame shift. And the score goes from 0 to 1, and we're very close to 1, showing very high constraint with only one loss of function described in the general population. It is also constrained against missense variation, as you can see with the Z-score that is very high here. And then, of course, the question is there clinical significance to this finding? And we also use another resource called HGMD, which catalogs a lot of the variants that have been described in the gene. And in looking here, we are seeing this one entry with a paper from 2020 in an individual with macrocephaly, intellectual disability, and sleep apnea. So of course, this looked very interesting. In looking at that paper, we can see that they actually had a de novo variant in a patient with these features. And in addition to that, they actually pulled a lot of cases, so 25 cases from the literature that had shown a deletion that overlapped with the locus where this gene is. And you can see here, the gene is right in the middle of this diagram. And this gene is deleted in all of these patients. And so they collected the different features that are seen in this patient and all the microdeletions. And I have highlighted in yellow here all the features that also overlap with what we're seeing in our patient. So it was, of course, looking extremely compelling evidence. And again, a reminder, this paper was online late 2019, so well after our clinical analysis had taken place. This was further supported by additional patients within the Decipher database, with both deletions here listed on the left, as well as some de novo small variants from the DDD project. And this was very compelling. So as part, again, of our data sharing efforts and collaboration within the rare disease community, we added this gene as a strong candidate into a website called Gene Matcher, which is intended to connect researchers and physicians that have a single patient with a rare disorder with other patients. And here is the link. This is the web link to Gene Matcher. And through this, we were actually able to find additional cases led by Dr. von der Lippe. And we were able to recently publish this finding. In this publication, we actually had a total of 12 new cases that have variants within this gene and overlapping features, which of course includes all the features that I had previously described in our patient. And this was a really exciting finding. And this is, of course, what we are hoping to achieve with all of our unsolved patients. So this is an example of where we found a strong candidate and were able to collaborate with others to find an answer. The next patient I would like to present is actually a patient that we were able to solve by the addition of other technologies. So here we have a proband with failure to thrive, hypotonia, persistent global developmental delays, and epilepsy. And family history was completely negative. I just wanted to mention that this pedigree is also from PhenoTips, and you can actually extract a beautiful figure like this from PhenoTips, which is very helpful. And so for this patient, we actually did not see this through the standard exome sequencing, but when you do genome sequencing, which has uninterrupted coverage, you can clearly see at the top here that the proband has a dip in coverage, which is consistent with a deletion of this gene, CACNA1A. This deletion covers 3 exons and is absent in mom, and it's consistent with the diagnosis of autosomal dominant neurodevelopmental disorder and epileptic encephalopathy. So this was a really good one as well that we had, and we were able to provide answers to the clinical team. Another example of what another successful story was patient 3. This patient was actually diagnosed prenatally with hepatosplenomegaly, and this persisted after birth. There was a liver biopsy done at 6 weeks that was concerning for multiple abnormalities. The initial clinical testing revealed a single pathogenic variant in the gene NPC1, and this gene is known to be associated with Niemann-Pick disease type C. However, we only had this one variant, and this is an autosomal recessive disorder, so we would really expect 2 variants. And also to add complexity to the analysis, we did not have parental samples available. Given this finding, there was a biochemical testing done. So they measured specifically oxysterol, and this was abnormal. So this establishes the clinical diagnosis of Niemann-Pick disease, and it really allows the clinical team to start interventions right away, but the patient remained without a molecular answer. Through our research study, we were able to find a second pathogenic variant that happened to be intronic, and as I mentioned before, we actually did not have parental samples. So typically you would want to show the one is maternally derived and the other variant is paternally derived to confirm that they're on opposite alleles. But in the absence of that, we are actually able to use the high fidelity long read genome sequencing from PacBio to show that each of these variants is actually on a different allele. So with that, we were able to completely give a molecular answer to this kiddo as well. And this has implications, of course, not just for the child itself, but for the family in, for example, planning for future pregnancies and understanding their recurrent risks. Finally, this is actually the last example I will present today. Patient 4 was a proband that presented with a concern for skeletal dysplasia and completely negative family history. He presented with overall shortening of the limbs, a short neck without webbing, and some digital anomalies. And he had had a clinical genome in 2016 that had been negative. And because he already had a genome, it was decided that on our research study, we would pursue the long read PacBio high fidelity sequencing. In doing so, we actually detected an interesting variant. So just to situate you here, you see that there's 4 panels actually, and it repeats proband mother. This is just because the top 2 panels represent the long read genome sequencing, and the bottom ones represent short read sequencing. And in both of them, you are seeing this variant that appears to be homozygous in the proband and heterozygous in mom. And this variant is actually changing the start codon of this gene, B3GALT6. Now, you may ask or be wondering, oh, but we are seeing this in the genome sequencing. How come? Well, I will get to that soon, but just to touch on the clinical relevance of this variant, we have seen there have been other variants at the start codon for this gene that are associated with some sort of skeletal dysplasia that seems to be variable, but it would be consistent with the presentation for this kiddo. So really, I just want to take a moment and go back to my slide and point out something that is not always thought of, but this analysis was done with the human build 38, which is what we are using for our research study constantly. And going back to the data that we had previously, which was build 37, there's actually a gap in coverage here. There's very, very low coverage. Even the top panel here is genome sequencing for this proband, and you can see very little coverage, but you do see 2 reads here with the arrow, and they do show the variant there. But with 2 reads, this would have been discarded as not compelling variation. And also at the bottom here, you can see mom as well as 2 other controls with exome sequencing. This part of the exon for this gene is completely not covered. So this was not detectable with build 37. When we looked further at why this may happen, it actually showed up as a known problematic region, which is difficult to align. And this was actually captured by ENCODE in sort of what they call their blacklist, which represents regions where the genome assembly is causing errors, and we are not seeing the coverage that we're hoping and can miss variation. And again, this is not something that might pop to to the top of the list is something to keep in mind, but it is something that is really important as well. And as expected, this alignment issue was corrected in Build 38, which is why we were able to see that variant come through in our analysis. Nonetheless, just to point out that exome sequencing will still have a gap in coverage in this region, but with genome sequencing, with Build 38, this would be captured. And so we were able to provide an answer to this kid that had been waiting a long time since that genome in 2016. So with that, I hope I was able to illustrate how we are incorporating a lot of information and how helpful PhenoTips has been in us reaching diagnostic answers for some of our kids, and that I hope that the examples I presented today were able to illustrate different ways that we are getting at those answers. As I mentioned earlier, this involves a very large number of people, which starts, of course, with the patients and the families that are agreeing to participate in the study, and we are so grateful for their participation. The Genomic Answers for Kids core team, which really works in all directions, from the technology to the recruitment to the analysis and everything in between. The clinical teams that are referring these patients and informing them of our study as well. Of course, our collaboration with PacBio, and last but not least, in any way, our collaboration with PhenoTips and how helpful they have been in supporting our endeavors. I'd like to thank everyone involved, and I'd like to thank you for listening. And I am happy to address any questions. And I also just want to leave here my direct work email address in case you have any questions. You're also welcome to contact me directly.
Thank you so much for the presentation, Anna. That was really, really interesting. I'm sure we will have a few questions coming in from the audience already, so we'll jump into those briefly. I actually have a few questions as well. Could you maybe talk a little bit more about what the inclusion criteria were for patients to participate in the study?
Yes, absolutely. So we are keeping the criteria really broad on purpose. We do not want to exclude anyone that could benefit from the study, but also, as I mentioned, this is sort of effort that goes beyond just each patient individually. We are creating we are concentrating a large amount of data that is going to help other patients within the study, but also beyond the study. So the only criteria,
do you know quality
individual. Excuse me.
Oh, sorry. I think there might have been a sort of a gap there in the internet for a second. Please continue.
So I was just saying, typically the proband will be someone that is 18 or younger and that has any constellation of phenotypic traits that are suspected to be of genetic origin. And then, of course, as we recruit the patient that is typically pediatric, we might recruit additional family members. So that would include adults that are affected within those families as well.
Thank you. I also wanted to ask, do you know or have any insights as to sort of across this patient cohort, what were the sort of average number of phenotypes per patient? And did that have any impact? And also, would you happen to have any information about the quality of the phenotyping or some of the clinical and family history information, and whether there was any correlation there to the outcomes of the study?
Yes, that is a great question, and it is somewhat complex. So as I mentioned earlier, when we were looking at HPO terms, there's different ranges of terms, and there's different specificity of the terms. So if we include a term like abnormality of the head, that is, of course, going to encompass a lot more people than if we include a term that is macrocephaly. That's already a lot less, but it's still really broad. So depending how specific the terms are, it might overlap more patients within our study. We did, however, try to look at whether numbers correlated with diagnostic outcome, and throughout the different sort of buckets. So if we were to divide those with a clear diagnosis, those with a that we have a candidate for, but we're not quite there yet, and those that we really have no clue of what might be causing their symptoms. For all of them, the average number of HPO terms that we end up with are between 6 and 7. And we're not seeing any correlation between the number, just the number, and the outcome. I will say we also, because of the sheer number of people we have involved in the study, how that information is captured is also variable based on who is doing it. It could be anyone from the recruitment team, or it could be the expert, this morphologist that saw the patient in clinic, and we're also not seeing a correlation there. That would be at least significant in a way that we could say that there's specific numbers or combinations to use that would more likely give you a diagnosis.
Mm-hmm. So we have some questions from the audience. We have a question asking, would the chromosome 19, 13.3 deletion be picked up by microarray?
A deletion of that region, I would imagine that many of those would, and given the literature review that they did in that specific paper, it is clear that some of them would. I cannot tell you the exact resolution of how small of a deletion in that region would be picked up by microarray, because I haven't myself looked. So it really will depend on the size of the deletion, but some of them would for sure.
Mm-hmm. We have another question here. What has surprised you in sequencing the first 5,000 to 6,000 participants? Were certain types of variants more common than others?
That is wow, that is a question I haven't heard before, but I appreciate it. And it is not easy to address that question, simply because me and the other people that are doing the analysis, we typically start the analysis on a patient-by-patient basis. And so when you're zooming in on a patient, you're really looking at what variation that specific patient has, and that appears to be pretty constant, with, of course, the caveat that if you remember earlier, we are filtering against common variation that's in public databases. So we do see a very large difference in number of variants that are considered rare, depending on the ethnicity of the patient, given that some ethnicities are very underrepresented in these populations. However, I would like to say from specifically our long read genome data, we are seeing some structural variation that was perhaps previously underappreciated, given just the fact that most of the sequencing is done with short read technology. And we did try to address some of this in our paper by specifically focusing on the variation that was inherited. So if we were seeing it in the proband and one of the parents, we believe that it was likely a true signal rather than just a noise from the sequencing or from the analysis, due to the lack of public resources for such types of data, we are seeing that there are structural variants there that we have not been capturing in the past and will probably require a lot of investigation and might provide some of the answers that we are missing.
Thank you for that. We have another question from the audience. How impactful was the collection of family history? Did it improve analysis in most cases?
Oh, so family history is really critical, and it is, I feel it's one of the oldest things that have shown to be powerful in establishing a genetic, a genetic origin of disease from, the oldest days of genetic descriptions and papers up to now. The family history really helps us both include and exclude variants. So if we have a variant that looks really compelling, but it's not segregating with the phenotype within a family, it becomes less compelling, not necessarily excluded, because then, of course, we have to consider other variables, including penetrance and expressivity, which means, the correlation whether having the variant necessarily means you have the phenotype. There are complexities like that, but family history, and specifically the larger the family that we recruit, the better it is for giving us the causal variant. With that, just wanted to add as well that for a lot of these rare disorders, it has been shown that there is a very high rate of what we call de novo variants. So it's a brand new variation that occurs in just a patient. And so for that, knowing that both parents are unaffected and having, just a child with both parents, that's already a very, very powerful way to do our analysis.
Thank you for that. We have another question from the audience from Michelle Fox. Since this is a research project, do the patients identified need to have findings confirmed in a clinical lab to take advantage of treatment, management, or clinical trials?
Yes, absolutely. So that is a very important question, and I thank you for bringing that up. So we actually have our research study, and the research lab sits directly next to the clinical lab. And the workflow that we have set up is that anything that is discovered on the research side and is found to be of clinical significance. And for that, we use the American College of Medical Genetics guidelines to determine clinical significance. Anything that is of clinical significance is then confirmed by an orthogonal method in the clinical lab and is reported into their clinical chart so that the physicians can act on those results.
And another audience member has actually asked, do you have access to the whole written medical record or just the HPO codes for each patient? And I guess that potentially plays into what you spoke about before about the clinical team at Children's Mercy also recording some of that data directly in PhenoTips.
Yes, absolutely. It absolutely ties together. So the vast majority of patients enrolled in Genomic Answer for Kids are children that are seen within the Children's Mercy Health System. And for those, we do have access to their complete chart. And we try our best to summarize that with the accurate HPO terms. However, we will go back to the chart if there is novel variation that seems potentially interesting, but that is not an exact match to what the HPO's terms are describing. And so we do go that one step further to look at the chart. And as I did mention earlier, if for those physicians and other clinical members that are using PhenoTips to record that information, we are able to just carry through and transfer to the research side. We are working with all subspecialties within the hospital. So not all subspecialties are using PhenoTips, but specifically genetics, they have really embraced using PhenoTips. However, we do have patients that are enrolled in our study through other collaborations that are not within Children's Mercy. So for those, we get a mixed bag. It could be anything from a detailed, clinical letter to just a very few HPO terms.
Mhm. We have another question from the audience. So Francis Sansbury is asking, good to hear your perspective regarding ZBT-V7A. Would you like to say more about using Decipher, Gene Matcher, Matchmaker Exchange for finding similar cases? Are you depositing your variants as opposed to genes of interest into one of those databases?
So that is a great question that touches upon how important all these resources are. We are currently only depositing the genes themselves into Gene Matcher. We have, last time we checked, we had over 160 genes in Gene Matcher from our study. I bet it's more now. But we are also sharing those genes through this public PhenoTips interface we have. However, I will say we are very quick to share what the variant is and with specific information about the transcript whenever we are communicating through these channels. And we have found that is important sometimes to clarify we're talking about, the same transcript of the gene, and specifically that there is phenotypic overlap between the patients to share the variant. My personal feedback that I've gotten from these collaborations is that knowing the variant ahead of time has not been particularly helpful, but I could be wrong and that could change. However, I think the success of Gene Matcher has proven that just sharing through the gene can get you really far.
In the last few minutes, can you speak a little bit to the criteria that will be assessed as part of providing access to the genomic answers for kids data set through PhenoTips, to the publicly accessible version?
So the reason that there is an extra layer to get access to the data is not because some people will receive access versus others based on who they are. Is simply we want to track these potential collaborators so that we can communicate on potential successful outcomes for these. And also we want to make sure that these are going to, research entities rather than perhaps commercial entities.
Certainly makes sense. So thank you, Dr. Cohen, and a big thank you also to our audience for joining us today and for all of the great questions. You will receive a link in your browser when this webinar ends, as well as an email that you can answer a brief survey if you missed the link. The email you receive after the webinar will also include a link to the PhenoTips Speaker Series page, where you can sign up to receive alerts on upcoming sessions. You can also visit phenotips.com and go to the resources tabs where the speaker series is available in a drop-down menu. All of the installments are available to stream on demand. In addition, the email you receive after this webinar will also include a link to register to our February webinar, which will be taking place on February 8th. The title of that webinar will be Improving Patient Experience in Clinical Genetics. It'll be featuring geneticists and quality improvement innovators, such as Dr. Ian Campbell, Dr. Bimal Chaudhary and Dr. Ankit Kumar. If you're interested to learn more about PhenoTips or collaborate on projects with us, you can send us an email to hello@phenotips.com. Looking forward to the next. Take care, everyone. Thank you very much.